Noswaith a croeso. Good evening and welcome everyone. And for our worldwide audience, kia ora atamari. So this evening's lecture has been chosen to coincide with World Diabetes Day, which falls on Sunday, 14th of November. And we're fortunate that we have not only one speaker, but two speakers this evening. So I'd just like to introduce our first speaker is going to be Professor Susan Wong, who is currently Professor of Experimental Diabetes and Metabolism at Cardiff University, as well as being an honorary consultant physician in diabetes at the University Hospital of Wales. Susan has many years of research into the causes of type 1 diabetes, as well as clinical experience in treating all aspects of diabetes mellitus. She considers that activities that engage and involve the public in understanding scientific research and educating people who do not have a medical or scientific training about medical issues to be extremely important and integral to her work. In 2018, Susan was awarded the Dorothy Hodgkin Lecture Award, an award that is given to a person internationally recognized for their contribution to basic research in the field of diabetes. So welcome, Susan. Our second speaker this evening is Dr. James Pearson, who is an early career researcher studying how white blood cells of the immune system cause the development of type 1 diabetes, with the aim of developing new areas of research that can lead to either preventing the development of type 1 diabetes or improving the lives of those with type 1 diabetes. His current research focus is to have an understanding of how the time of day changes our immune cells' ability to communicate with the insulin-producing beta cells in the pancreas that are targeted and destroyed in type 1 diabetes. He's currently funded by a Medical Research Council Career Development Award and a Juvenile Diabetes Research Foundation UK grant. So welcome, James, as well. Over to you, Susan.
Thank you very much, James. And thank you very much, everyone, for joining us tonight to celebrate the centenary of insulin discovery. I'm going to share my screen. So tonight we're going to talk about what insulin is and why it's important. We're going to discuss the scientific discoveries related to insulin over the last 100 years. This has considerable relevance to diabetes, so we will talk about what that is, and why the discovery of insulin was so important to people who live with diabetes. And finally, for type 1 diabetes and research, we're going to think about the future. And just as a disclaimer right at the beginning, if either I or James say we in this talk, we're talking about being members of the scientific research community, because we're not going to talk directly about our own research. So firstly, what is insulin? Insulin comes from this organ, the pancreas, which is very deep in the body and sits behind the stomach. Most of the pancreas is made up of cells that produce chemicals, enzymes to help us digest food. But within the pancreas, we have islands of cells called the islets of Langerhans. And these contain beta cells that produce the hormone, and these are chemical messengers that is called insulin. And these islands don't make up very much of the pancreas, but the hormone insulin that they produce is really very important. And you can see here one of these islands, and the insulin within it is stained in green. So we have these islands in the pancreas. When we eat food, and particularly the group of foods that we call carbohydrates, such as potatoes or bread or rice or other grains, these are digested in the gut, and then glucose, which is a product of the carbohydrates, is released into the blood. And when the blood circulates through the pancreas, This stimulates the production of insulin. Insulin acts on the tissues which use glucose. The muscle is a very important tissue that does this, but in fact, many of the tissues in the body also use glucose, and they require insulin to help the glucose to get in. So glucose will bind to receptors on the surface of here shown a muscle, and they signal inside the muscle, and this brings to the surface transporters to which glucose is attached, and this helps them to go into the muscle cell. And the glucose inside the muscle cell provides energy, as it does in other tissues. But glucose is also stored not only in muscle, but also in the liver, and when it is in excess, it will also get stored in fat. So why are we celebrating 100 years since the discovery of insulin? Diabetes mellitus, which means sugar in the urine, has been recognized as a condition since ancient times, as far back as Hippocrates. So that's nearly 2,500 years ago. And it's not just one condition. And people have been studying the causes of this for quite some time. So in 1889, there were two German scientists, von Mering and Minkowski, who had shown that the pancreas contained substances that were able to regulate blood glucose. So they had known that the pancreas had a key to carbohydrate metabolism. But they didn't take that any further. In 1916, a Romanian doctor, Nicolae Paulescu, developed an extract from the pancreas which was able to normalize glucose in a dog with diabetes. So he might have been someone who could have discovered insulin. But his discovery was made in the First World War, and he wasn't able to take these studies any further at that time. Before 1921, children who had diabetes, which is now known to be type 1 diabetes, which is highly dependent on insulin, inevitably died. The children looked as if they were starving, and in terms of their physiology, that's exactly what they were having, starvation, because although there was glucose around in the bloodstream, they were not able to use it. And the only known treatment at that time was a starvation diet, which wasn't effective for very long. So why is 1921 so important? In that year, Frederick Banting, a surgeon, together with Charles Best and Marjorie the dog, who had diabetes, showed that after injection of Marjorie with a pancreatic extract, Marjorie's glucose reduced towards normal. This is one of the early machines at the University of Toronto where this study was done that was able to extract this principle from the pancreas. And this extract was in a form that would then be taken forward for the development of a therapy. So this was really important because the extract, which they found, was further developed into something that could be therapeutic. So they went on to work with James Collip, who was a biochemist, and they were able to extract something that could be injected into humans. And this is the first person who was injected with this pancreatic extract in 1922. Leonard Thompson was 14 years old, and he lived for a further 13 years beyond this and died of pneumonia. This is the same girl that you saw earlier, and this was before insulin, and this is what she looked like 4 months after being treated with insulin. And this is the same for this young man who was very small, and he looked quite different after treatment with insulin. So this really was life-saving treatment. And so the Nobel Prize in Physiology or Medicine was awarded to Banting and John McLeod, who was head of the lab in which Banting did his studies, and they were awarded this for the discovery of insulin in 1923. Unfortunately, as with many important scientific discoveries and prizes attached to this, there was some acrimony about this. And Frederick Banting shared his Nobel Prize with Charles Best, and John MacLeod shared his Nobel Prize with the biochemist James Collip, who undoubtedly played a very important role in being able to extract the principle from the pancreas in order that it could be injected. And from that time, it was taken up by two pharmaceutical companies, firstly, the Lilly Company in America, and then the Novo Nordisk Company in Denmark, and was taken forward to make insulin for therapeutic use. And insulin has undoubtedly saved lives of many people who had diabetes. At the beginning, it was given with glass syringes with metal needles, and these needed to be sterilized either by boiling or keeping them in spirit. And these needles were used many times and often became very blunt. As with people who have type 1 diabetes today, there was a lot of kit attached to having diabetes, and people would need to carry their syringes, needles, and some reagents to test urine, and I'll come back to that shortly. So let's think a little bit about what insulin is. Whenever we have a scientific discovery of this type, many important questions come up. And this life-saving extract was the subject of intense investigation. Frederick Sanger in Cambridge won the Nobel Prize for Chemistry in 1958 because he discovered that insulin was a protein, and he discovered what that amino acid, the building blocks of a protein, exactly was. There's a B chain, which is 30 amino acids, linked to an A chain, 21 amino acids, joined by another chain, which is cut off before insulin can be active. And this was important because insulin, in its native form in the pancreas, acts only for a very short time. So if it's injected, people need to take lots of injections. And one of the important things about understanding the nature of such a protein is that scientists have been able to alter some of the building blocks to develop different types of insulin that can last for a longer or shorter time in the body. But the other thing about insulin in those early days was that it was extracted from the pancreas of cows and pigs, and every preparation was slightly different because it was very difficult to standardize the insulin because the measurements were not very accurate. So another Nobel Prize was awarded to Rosalind Yallow, who, with Solomon Burson in New York, used a test called a radioimmunoassay to be able to measure small amounts of insulin accurately. And they did this work in 1960, and she shared the Nobel Prize in Medicine in 1977. So from this time, people were able to measure very small amounts of insulin. And although this was important for the preparation of insulin and the amount that people needed to take, it was also important because from that time, insulin could be measured in the blood, very small amounts. And one of the discoveries that they made was that some people with diabetes didn't have a complete lack of insulin, but rather the insulin that could be measured seemed not to be very effective because they could actually measure the insulin levels. And this is the type of diabetes that we now call type 2 diabetes, which was previously called non-insulin dependent diabetes. There were still some questions about what insulin looked like. How was it put together? Dorothy Hodgkin, working with colleagues in Oxford, had started work on this in the 1930s, and they took X-ray photographs of insulin crystals, which look like this. But she continued to work on this for over the next 30 years, and they discovered that actually insulin was put together as 6 chains together with zinc in a crystal. She had won a Nobel Prize early in the 1960s for the discovery of vitamin B12, but for this discovery, she was honored with a commemorative stamp. Understanding the structure of insulin has helped scientists to find out how insulin works. So, lots of discoveries related to insulin itself. But we've talked a little bit at the beginning about the fact that if people don't have insulin, this leads to this group of diseases that we call diabetes mellitus, sugar in the urine. There's not one disease, however, and people who live with diabetes have a number of different types. And so it's important to understand that it isn't just one condition. Type 1 diabetes affects about 10% of people who live with diabetes, and that's about 400,000 people in the UK. And this is what was previously called insulin-dependent diabetes. People who live with this have insulin deficiency. Their pancreas does not produce enough insulin for the body needs. And in some people, after some time, insulin can't be measured at all. And this is due to an immune attack on those insulin-producing beta cells, and I will talk some more about this shortly. The most common type of diabetes is type 2 diabetes, which affects about 85% of people living with diabetes, and that's about 4 million people in the UK. This was previously called non-insulin dependent diabetes. We know that people who have type 2 diabetes have some insulin deficiency, but insulin can, at least in the early stages, be measured. And it seems that there is not enough insulin for the body's needs. But the insulin that is produced doesn't function as well as we might expect, and it is not therefore stored efficiently in muscles and fat. And this is what we call insulin resistance. There are several other types of diabetes, which include gestational diabetes, which occurs in pregnancy but goes away later, but may be a risk factor for type 2 diabetes, diabetes that occurs with cystic fibrosis, and much smaller numbers of people have something called monogenic diabetes, which is a genetic type of diabetes. So why do people get diabetes? We know that type 1 diabetes is not inherited. People inherit genes which make them more or less susceptible to type 1 diabetes. This discovery was first made in the 1970s. The most important genes involved in genetic susceptibility are what we call human leukocyte antigen genes, HLA. These genes are very important for the normal function of the immune system, and particularly the function of white blood cells. And they are the most important susceptibility genes relating to type 1 diabetes. We now know that there are more than 40 other genes that contribute some risk, but none of them as much as the HLA genes. We have 23 pairs of genes which have many, many DNA molecules. And if we look at the susceptibility to type 1 diabetes, we can see, if we think about how much that contribution to the susceptibility is, the HLA contributes this much, and the other genes much less. It's interesting that most of the other genes involved in susceptibility to type 1 diabetes are those shown here in the blue-green bars. And interestingly, they mostly code for immune functions. The genes involved in susceptibility to type 2 diabetes are quite different from those involved in type 1 diabetes. The genetic susceptibility factors and understanding these will allow people at higher risk of developing type 1 diabetes to be identified, and we have recently developed genetic risk scores. This will allow people to be identified who can most likely benefit from preventive treatment in the future. The other thing that told us that type 1 diabetes was related to the immune system was the detection of autoantibodies to components of the islets in the pancreas. Now, you will have heard about antibodies in the context of infection, and antibodies are a marker of immune activity. And in type 1 diabetes, these show immune activity against the insulin-producing beta cells. The antibodies are produced by white blood cells called B cells, and they're found on the surface, and they're also released from these B cells into the blood. If we take a component of the blood called the serum, and we put this onto sections, very thin slices of pancreas, and then do some staining, you can see here that those antibodies are shown up as yellow, and this lights up these insulin-producing cells. This isn't seen in people who don't have type 1 diabetes. Why is this important? We know if there are 2 or more different types of antibodies against insulin-producing beta cells in the bloodstream at a high level, this indicates an increased risk of developing type 1 diabetes. But we know that the B cells themselves don't attack the insulin-producing cells. So what is it that's causing that damage? In the 1980s, the group of white blood cells called T cells had intense investigation and discovery. And it was shown by a very careful Scottish pathologist, Alan Fowlis, that when sections of pancreas were examined under the microscope from people who had died having had diabetes and donated their pancreas, that you could see in these islets, which are ringed here in orange, that both around the edge as well as inside, there were some dark staining cells, which turned out to be these white blood cells called T cells. And again, this is something that you don't see in people who don't have type 1 diabetes. These T cells are normally very important for helping fight infection. And in diabetes, they're here in the wrong place because they shouldn't be here within the islets. So from this point, it was understood that the damage done to the insulin-producing cells was likely to be done by these T cells. And the fact that we know that T cells enter the islets in the pancreas in type 1 diabetes is important because now we have a target to think about seeing if we can prevent them from going in and develop immunotherapy against these T cells in type 1 diabetes. But aside from all the scientific discovery, right from the time when insulin was discovered, we know that people who live with type 1 diabetes must be able to manage blood glucose levels, because if it is too high, people feel very ill. And it's important that they are able to manage glucose levels within a certain range. In the 1990s, there were 2 large trials to see whether it was indeed important to try and control blood glucose levels. The Diabetes Control and Complications Trial, DCCT, for type 1, and the UK Prospective Diabetes Study, UKPDS, for type 2 diabetes, were done to see whether high blood glucose levels made a difference to future development of complications, as earlier it had been shown that people who seemed to have very high levels of blood glucose over a long period of time had more risk of developing eye, kidney, and nerve disease. And these trials shows that blood glucose does matter for both type 1 and type 2 diabetes. But one of the problems is that if the blood glucose is controlled too tightly and frequent and severe low blood glucose occurs, this is also not good because the brain and the heart both need glucose for best function. And so, in fact, it's a balance. And what we try and do these days is help people to manage blood glucose within a range that's not too high, but also not too low. And in addition, monitoring and controlling high blood pressure control, cholesterol, and very important and very crucially, people should not smoke for best health in people who have any type of diabetes. So how is insulin given? We saw earlier that we have glass syringes, and in the 1960s, plastic syringes and disposable needles were introduced. These were certainly sharper and much less likely to be made very blunt by repeated use. There were also other contraptions, which include this Palmer injector, which was developed by a Scottish farmer who really hated giving injections of insulin for his diabetes. And although this looks like a very fierce gun-like contraption, I have heard firsthand from a couple of people who've used this that actually it was more comfortable to use this rather than the insulin in the syringes. These days, we have much more convenient ways of giving insulin with a pen device that has glucose loaded in, that has insulin loaded into it, and the needles have become very much smaller. This is the size that might have been used much earlier, and they have become progressively thinner and shorter. And so giving injections with these much smaller needles is certainly much less painful. There has also been a needle-free jet device developed, but I'm told that this actually is more painful than some of the smaller needles. But the other advance has been in the development of insulin pumps to help with delivery. Dr. Arnold Kadish in the 1960s developed this prototype of this huge machine, which I don't think was ever introduced into clinical practice. But certainly the earlier pumps worked on clockwork, and they were also potentially the size of bricks. These days, there have been much greater advances in this, and we have insulin pumps that are smaller than mobile phones. They have a computer inside, they take insulin, and they're worn on a belt, and the insulin is delivered through a fine tubing into a little tube underneath the skin. And when people need to eat, they still need to calculate how much they want to eat in terms of carbohydrate, and then feed that information into the pump together with the blood glucose, and the pump will calculate how much insulin needs to be given. But those attention to details in terms of what is going to be eaten still needs to be done. There are other pumps. This is a patch pump, and it's into the skin. There are no tubes, and it's controlled by a handset that works a little bit like a mobile phone. Testing of glucose is important because actually not only is it important to know what is going to be eaten, but also what the blood glucose is. And as I've said, it's important that it is controlled within a range. Early on, before 1940, only urine could be tested, and a chemical test called a Benedict's test was done. Later on, there were tablets and then sticks. But as Someone who has lived with diabetes for longer than 50 years said to me recently, you didn't really need a urine test to tell you that your glucose was high because you didn't feel very well, if that was the case. Blood glucose sticks didn't appear till the 1960s. And in the early days, the first machine was very clumsy and it weighed more than 2 pounds. These days, there's a huge variety of blood glucose testing meters. Many of them test tiny amounts of blood, and most meters are smaller than mobile phones. This one is about the size of a mobile phone, so you can see some of the others are very much smaller. A great advance over the last 10 years has been the ability to test skin glucose. So this is not a blood glucose test, but skin glucose reflects the blood glucose, but can be a few minutes behind. Many people will have seen some wearing this flash glucose monitoring. It's a disc on the arm that measures skin glucose, and when this scanner is put over it, it will record the skin glucose. There are other types of this continuous glucose monitoring, and they produce a trace over 24 hours that we can see exactly where it has been through that period. And we try and encourage maintenance of that glucose within a certain clear range, which is not too high and not too low. But perhaps one of the greatest advances has been that we have moved much closer to the artificial pancreas. Here we have a glucose sensor which measures the skin glucose, and it can be combined with an insulin pump. Because at the end of the day, what a pancreas needs to be able to do is to be able to sense a glucose level and to deliver an appropriate amount of insulin. So the glucose sensor will sense the glucose in the skin, and it will transmit the information to the pump, and the pump can deliver insulin. Now, this doesn't work completely automatically because, again, it will work on a background level. When someone wants to do exercise or eat, again, the same thing has to happen, that there needs to be a calculation and an adjustment that is made manually so that the best levels of glucose in the blood can be achieved. Many famous people have lived with type 1 diabetes, and it has become much easier over the last 10, 20, 30 years. We can see Olympic athletes, actors, singers, models. Theresa May, our former Prime Minister of the UK, has type 1 diabetes, as Sonia Sotomayor, the Supreme Court Justice of the USA. So we have ways of giving insulin, and undoubtedly it is really important for maintenance of life in people who have type 1 diabetes. But as we go into the future, we would like to make things even better, if possible. And because it's really difficult from the outside to maintain the same control of the blood glucose as it is from the inside, one of the things that we would like to be considering is whether we can replace the function of the insulin-producing cells. We know that in type 1 diabetes, many of the insulin-producing cells have been damaged by the immune system. In some people, there is still a little bit of insulin, but in many, there isn't very much at all. And both T cells and B cells are involved in this attack. So in terms of the science going forward, could scientists replace or restore the cells in people who live with type 1 diabetes and then prevent it from happening again? Could we prevent this from happening in people who don't have diabetes but are at risk? So we have two components. If the insulin-producing cells are damaged and not there, they need to be restored or replaced. Could we do this by transplantation? And the answer is yes. People have had islets or pancreas transplants, and this is to some extent successful. The problem is that over a long term, these are not genetically matched, and people need to take drugs that suppress the immune system to prevent rejection. The other question is, could new beta cells be made? Could they be made from stem cells? And there is also a lot of research going into this. Could new beta cells be made from embryonic or pluripotent stem cells, cells that haven't yet decided what they want to be when they grow up? Could the beta cells come from the few beta cells that might be left to divide? This is something that is of interest, but hasn't had a tremendous amount of success in humans. Could new beta cells be made from cells that normally have other functions in the body? So could other cells be reprogrammed? Then there are even more experimental types of stem cells or islets that could be formed. Could there be engineered islets in pigs, or could human pancreas be grown in animals? But as mentioned, this is very much more experimental. And currently the most promising is new beta cells from stem cells. But even if we can replace those beta cells, the other side of that problem is that we would need to protect them from being damaged by the immune system again. And so I'm going to pass over now to James to tell you a little bit about this. James.
Thank you very much, Susan. I'll just bring up my screen. There we go. So as Professor Wong mentioned, the T cells are involved in the destruction of those islet beta cells in the pancreas. But interestingly, all those T cells in our body, they have a specific marker on their surface that we call CD3. And this means that we have a way to identify the T cells and to actually target them for therapy. And we can do this using an antibody that we call an anti-CD3 antibody, which recognizes that CD3 on the surface of those T cells. In clinical studies using this anti-CD3 antibody, what we found is that it causes those T cells to actually go to sleep. It turns them off. And it does this through a process that we call T cell exhaustion. And this means that those T cells in turn then are unable to attack and destroy those islet beta cells, meaning that it's protected those islet beta cells and allowed them to continue producing insulin. This therapy has been administered to people with type 1 diabetes. So if we've mentioned, insulin is very important as a readout to tell us that those islet beta cells are not being damaged. So the more insulin that we're able to detect, so at the higher level, the more likely those islet beta cells have been protected from the T cells destroying them. You can see in this study of individuals with type 1 diabetes that a group of individuals, around 3 out of the 10 of them, had detectable levels of insulin. However, when we give individuals anti-CD3 treatment, what you can see is that you've almost doubled the number of people that have detectable levels of insulin in their body. And therefore, this suggests that the treatment with anti-CD3 helps to protect those beta cells from destruction. Importantly, anti-CD3 therapy has also been given to individuals who are at high risk of developing type 1 diabetes, but have not yet developed diabetes themselves. So on this graph, what you can see is that we've got a proportion of people that do not have diabetes. So if the line stayed at 100, this would mean that no one has developed diabetes. Whereas if the line drops down, it means that more people have developed diabetes until 0, where everyone in the study would have developed diabetes. And this is a time since therapy administration on this axis here. What you can see in this black line is that those individuals that didn't receive any treatment, by around 5 years post-treatment, we're looking at about 80% of people that have been diagnosed with type 1 diabetes. However, in those individuals given anti-CD3 therapy, what you can see is that about half of those individuals are still diabetes-free at the point of around 5 years of post-treatment. Importantly, this also shows, as there's a difference between the black line and the red line, this gap in the middle really suggests to us that this treatment also delays the development of type 1 diabetes in individuals at risk of developing disease. Another way to prevent those T cells from being destroyed by the islet beta cells is to target inflammatory molecules that the T cells produce. And in this case, we're talking about cytokines. These cytokines can be produced by the T cells and recognized by those islet beta cells, which lead to their destruction. Therefore, identifying ways to stop those cytokines reaching the islet beta cells, for example, through an anti-cytokine antibody, would make sure that those islet beta cells can be preserved and can continue to produce insulin. In a study of children and young adults with type 1 diabetes, the researchers wanted to know if their anti-cytokine antibody could actually preserve and protect those insulin-producing beta cells. So what you can see on this side is, we're looking at this case, a change in insulin, where the higher the line falls on this axes here, means that there's more insulin being produced. However, as you go further down here, we're getting less insulin, meaning that there's been a reduction in the amount of insulin produced. And again, on this axis, we've got time since treatment started. So you can see in this blue line, this is our placebo or control group, where by around a year after therapy administration, those individuals have lost about half of their ability to secrete insulin. However, in those individuals that received the anti-cytokine treatment shown here in orange, you can see that around 6 months post-treatment, these individuals are still maintaining their level of insulin that they had at the beginning of the study. However, this slightly falls off, but still they have higher levels of insulin being produced than those that didn't receive therapy at all. This suggests that blocking those inflammatory cytokines may prevent the beta cell death caused by the T cells. Another way that we can help or target the immune cells involved in the destruction, as Professor Wong mentioned, there are other cells like B cells that can contribute to that process. In this case, the B cells can talk to the T cells and actively encourage them to destroy those islet beta cells to cause type 1 diabetes. Therefore, we know, because all B cells express one marker on their surface, a marker called CD20, we can actually develop ways similar to how we did in T cells targeting CD3. We can target the CD20 on those B cells specifically, which in this case, following treatment, actually removes those B cells from circulation, which prevents their ability to encourage the T cells to cause disease, which means then that you're protecting those islet beta cells from any T cell damage. In individuals with type 1 diabetes, researchers found that those individuals given anti-CD20 had changes in the amount of insulin being detected. So here on this graph, what you can see is that we're talking about the percentage of people with detectable levels of insulin. So 100, everyone in the study would have a detectable insulin level, whereas those down at the bottom end at 0 would have all the people in the study would have no insulin that's able to be detected. And again, we're looking at over time, in this case in months. And what you can see in this red line representing the placebo is that around 35% of people left in the study actually still had detectable levels of insulin at 30 months post-therapy. When you compare this to the blue line, which is our anti-CD20 treated group, you can see that this proportion is actually increased to around 65%. And therefore, this suggests that there are a number of options that could delay the destruction of the beta cells caused by the T cells themselves. So far, all of the strategies I've talked about protecting those islet beta cells are all focused on trying to dampen down that inflammatory situation that the T cells are damaging the islet beta cells. However, there's also an important arm of the immune system that controls any type of immune response, and that's controlled by cells that we call regulatory T cells. And these T cells, their main aim is to suppress and reduce any activation of the T cells that could cause disease. Unfortunately, those regulatory cells in people with type 1 diabetes just don't work very well. And therefore, finding ways that we can boost those cells to encourage them to stop the immune attack is obviously much needed. One way we can do this is to feed these regulatory cells with a low dose of IL-2, in which it encourages those regulatory cells to become much more active and engaged with the whole process taking place, which can lead to them stopping any damage to the islet beta cells caused by those T cells, and therefore would provide us with a way to make sure that the insulin production from those islet beta cells is preserved. In a study with children newly diagnosed with type 1 diabetes, they found after IL-2 therapy that there were two types of response. So again, in this graph, we're talking about a change in insulin, where more insulin at the top and less insulin at the bottom. And again, time following treatment on this axis down here. What they found was that there were a group of children who didn't really respond well to the treatment. And you can see this because we're getting quite a drop in the amount of insulin that's been detected. When we compare that to a group of children who did respond, you can see that after about a year of therapy, we're beginning to rescue and sort of keep that insulin level at the same level that it was sort of 6 months earlier. You can see that there's still a delay almost in this response taking place. This process, this therapy, is not a quick response and actually takes time before it's able to take over and help preserve those insulin-producing cells. But what this study told us was that IL-2 therapy may actually help prevent the beta cell destruction in some individuals with type 1 diabetes, and it's currently unclear which individuals may benefit the most from this type of therapy. The final therapy that I want to mention involves the role of germs in our body, the role that they have on educating our T cells. The role of T cells in our gut is to survey the gut to make sure that there's no damage or no infection taking place. However, in type 1 diabetes, these germs behave a little bit differently, and they can actually cause damage and invade our bodies to cause problems. When the T cell encounters these germs in places they shouldn't be, those T cells become switched on and become quite angry, which leads to them traveling around the body and then targeting the islet beta cell, causing destruction and damage. One way to prevent this from happening is through a process that we call a fecal microbiota transplant. And the idea of this is that we remove any bad bacteria and we can replace it with good beneficial germs that can help protect our barrier and make sure that the germs don't invade our body. This would mean that those T cells can travel around the gut to check everything's okay and realize that there's no damage there, meaning that there's no need to go to the islet beta cells and destroy them. So far, these studies have been very preliminary, but have actually shown some really important findings in that we can actually preserve the insulin production from those islet beta cells following a fecal microbiota transplant, often in the form of a capsule given to people during this study. I also want to talk a little bit about those at risk and how we identify them. As Professor Wong mentioned, that there are those individuals who have a genetic risk, and we're able to identify those quite easily looking at the genes and the DNA that they may have inherited from parents. Professor Wong also mentioned that there are markers of the immune system that we can detect in blood that provide us with a readout to tell us that there's an ongoing immune problem in the pancreas. And these are these autoantibodies. There's also potentially a role for the type of germs that live in our body, given that they may cause some damage to our intestine. We can also potentially identify those and associate all of these things together to identify someone's relative risk of developing diabetes. The problem is we don't currently have a mass screening program for this. So the only way we're able to identify anyone at high risk of developing type 1 diabetes is through people taking part in clinical studies. And therefore, we should take an example from Bavaria in Germany or Finland, who have these screening programs set up so that if a therapy becomes available, they can very quickly roll it out to those individuals who would most likely benefit from the therapy. In summary, what we've shown you today is that the discovery of insulin 100 years later is still saving millions of lives, and that there are some really promising therapies targeting the T cells that can cause type 1 diabetes and therefore hopefully in the future prevent the development of disease. However, as I've mentioned recently, we need to be able to identify those individuals who would benefit the most from any treatment. I'd like to say thank you very much everyone for listening. I'd like to thank our diabetes research group for the support that they provided and all the research that they do. I'd also like to thank our research funders and also all the individuals who have taken part in our numerous clinical studies. We certainly could not have done the work without any of them. And I'd like to just finally hand back to Professor Wong to give you an update on a very exciting World Diabetes Day project that's currently ongoing.
Thank you, James. So just to say that Sunday is World Diabetes Day, and we have had an exhibition at Llandoch Hospital in Cardiff from the 14th of October, and it will be extended until the 22nd of November. We recognise that many people can't necessarily come to this, and we have a website with a virtual exhibition there. And what we have in this exhibition are the viewpoints of many people, over 100, who have expressed a view of what diabetes means to them. We have a project that outlines the history, some of which I have told you tonight. We've also 2 commissioned artworks by Bridget O'Brien, an artist who has been working with our research group over the last 2 years. And so I hope as many of you as are near Cardiff as possible can come and celebrate World Diabetes Day and look at the exhibition. But if you can't, please do go and have a look at this online. Thank you very much, and thank you for your attention.
Thanks very much indeed, Susan, for that most authoritative overview on type 1 diabetes. And thank you, James, as well, for providing us with those exciting insights into potential new therapies for the condition. Please, would our attendees kindly put their questions in the Q&A for Susan and James? I see one's come in already, maybe for you, James. Is it possible to combine multiple treatments and therapies together at the same time to potentially further improve insulin levels?
Yeah, that's a really interesting question. It's certainly possible, but I think the issues that we've had previously is that we need to know how each individual therapy works first, and that we can get that optimized before we then conduct any sort of dual trials where we use combination approaches. It's certainly going to be something that happens in the future, I can almost guarantee it, but we just need to sort of optimize those individual therapies first.
Great, thank you. Perhaps one for Susan. Susan, if I could just come in, maybe you could dispel the myth. You've already touched on it briefly this evening. I'm a bit of a rugby fan, and I'm aware that the English rugby player Henry Slade is a type 1 diabetic. And also there's a road racing, a pro road racing cycling team, Team Novo Nordisk. So can you just, highlight again that, people with type 1 diabetes can very much live normal lives and to the extent of competing high level sport?
Yes, so people can certainly do all of those sorts of things. It is hard work. I think no one should underestimate the fact that people who have type 1 diabetes have to work much harder at it. But absolutely people can do elite sports, and there have been many people who do marathons, ultra marathons. There is a well-known Scottish athlete, and he's done the Marathon des Sables across the desert in Africa. And so undoubtedly people can. We encourage people to do exercise and take part in all of this, there are tips and ways of thinking about it, and it's really important to think about how to manage the insulin and glucose levels so that they're not too high and not too low for optimal performance.
Right. Thank you for that. And Susan, I've tended to think of type 1 diabetes as a disease of sort of young children, or certainly manifesting first in young children or in teenagers, adolescents. But clearly it can manifest at any stage in life, as we've seen with our former Prime Minister. And is there anything then that, in terms of the genes that predispose to type 1 diabetes, are they different in people who manifest it much later in life?
Well, that's a really interesting question too. In fact, the genetic susceptibilities are very similar. People absolutely can develop diabetes later on in life, and about half of type 1 diabetes develops in people who are beyond childhood. But one of the things that has been noticed is that people who develop diabetes later may have less of the susceptibility genes. So if there are many susceptibility genes and the ones that give the highest risk, those individuals are more likely to develop diabetes early. It's not to say that it can't happen later, but sometimes in people who develop diabetes later, they have more of the less important genes.
Right. Thank you, Susan. A question here in the chat, in the Q&A. How do our rates of diabetes compare with those of other countries? So I'm not sure if that's type 1 or maybe both types of diabetes.
Okay, so type 1 diabetes, we're not at the top of the league table. Finland has the highest rate of type 1 diabetes in the world. But the UK is quite high. So if we divide the world in terms of different rates of type 1 first, the countries of Northern Europe have some of the highest levels, but that also includes Australia and North America, and countries of Africa and Asia tend to have less type 1 diabetes. And this may be a combination both of genetic susceptibility as well as some of the environmental factors that James has talked about. In terms of type 2 diabetes, the rates are high in many places and they are increasing. And they're increasing at a rate that is higher probably in the less developed countries of the world. And again, this has to do with genetic susceptibility, but also changes in weight. It's not so much absolute weight that is important in type 2 diabetes, but weight changes. And as society has different lifestyles and different ways of operating in our civilized world, this has led to an increase in type 2 diabetes all over the world.
Another excellent question here. How does lowering T cell function against islet B cells potentially affect T cell function against other potentially harmful pathogens? Is that going to be a concern?
Yeah, it's a really good question that's raised. And actually, I think the current pandemic actually sheds some light on this in a lot of ways, because people with both type 1 or type 2 diabetes are more susceptible to severe COVID infections. So there is something that's ongoing in the immune system that does predispose some individuals to develop sort of an immune response that's quite poor to other pathogens. I think it's quite clear at the moment that we don't fully understand this. It links to some of the genetic susceptibility factors that Susan has mentioned. There are some genes that are sort of altered in individuals at risk in relation to responding to viruses and to bacteria, but it's not clear yet how those work together and what it could mean for sort of susceptibility to other diseases. But it's definitely a really good question and something that we hope we'll be able to sort of address moving forward with our research.
Is there a concern that with some of the therapies, James, that you will predispose individuals then to, pathogenic organisms?
Yeah, there was very early on during the development of these therapies, because obviously you're either sort of stopping T cells from working, or you're removing some parts of the immune system to stop them from sort of adding or encouraging the T cells to cause damage. So there's certainly a risk of sort of infection at those points, because you are a little bit more susceptible because you don't have a full complement of immune cells. But I think compared to the other options where, as Susan mentioned, you've got transplantation, which would require you to have a lot of immunosuppressives where your immune system would be almost shut down, we can quite easily, in a clinical setting at least, mediate some of these risks quite a lot. Susan, would you have anything to add to that?
So absolutely, that at the time of taking the therapy, there can be a short temporary effect on susceptibility to infection. But one of the aims of immune therapy for type 1 diabetes would be to very specifically target the cells that are doing the damage. And so we're not looking to generally dampen down the immune system. And indeed, this is a very important fact, that we would try to really avoid having a general immune suppressive function. And a lot of work is being done to try and find out whether we can very specifically target the few cells that do the damage. And one of the ways we try to do this is by knowing what those cells recognize, to use that knowledge of what they're targeting to try and turn those specific ones off, while leaving the rest of the immune system to do the really important job of fighting infection. And so in general, it would only be, if there is any effect, it would only be at the time of, very short time of taking the treatment. And then the expectation is that immune function would not be affected and able to deal fully with infection.
Thank you both. Celiac disease and diabetes may have some links. Is this related to the bowel effects you mentioned and the fecal microbiota transplant developments in any way? Maybe comment on that.
Yeah, so people with type 1 diabetes and celiac disease, they actually share genetic susceptibility. So as Susan mentioned, the HLA genes are really the most important genes that dictate someone's risk of developing type 1 diabetes. And that is shared with anyone who develops celiac. And I think around a third of people with type 1 diabetes will experience or get diagnosed with celiac disease. It's not clear yet whether the microbiota could influence or improve that situation, as far as I know at the moment, but it is certainly something that could be looked at.
Great, thank you, James. What is the percentage of type 1 diabetics actually using pumps, and why is this percentage so low?
Yes, so it varies across the country. Mm-hmm. Children, there are a greater percentage of children actually on pumps, as this is one of the treatments that is advocated for children. In adults, the percentage probably varies between about 15 and 25%. I would stress that although it looks great, and without doubt people can get some very good blood glucose control with pumps, this type of treatment isn't something that everybody actually wants. There certainly may be a waiting list to get onto pumps, because a lot of training needs to go into using pumps. And we have guidelines about the indications for using pumps. But actually, if everybody was offered a pump, not everybody would want to have one, because it is something that requires quite a lot of attention, even more to the food, the calculation of the carbohydrates, the watching of the glucose levels, because in a pump, all the insulin that's going in is short acting. And so there is no backup of a long acting insulin that could protect you if something was going wrong. And so when people consider exactly what it is that's involved with using a pump, not everybody would actually choose to have a pump. And certainly not everybody wants to have something connected to them and carry the extra paraphernalia that is required for that. It does vary around the country. We have had low use of pumps in the past in Wales. And so that is actually increasing. And it does vary around the country, but I would say that probably no more than 25% of people, adults I'm now talking about, would be using a pump. The percentage is quite a lot higher in children.
Okay, thank you, Susan. Going back to microbiota again, then, can diet help to improve the microbiota diversity in people with type 1, or maybe possibly delay the development of full-blown diabetes in individuals who are at risk?
Yeah, so diet can certainly change the bacteria and the germs that live in your body, and that can happen through multiple ways where it can change how your immune system works. So there is that potential link there that could be taken advantage of. In the early studies that were done in clinical sort of fields, changes to sort of the microbiota through changes in fiber, for example, weren't very successful. They didn't really show any difference in either the preservation of the insulin-producing beta cells or in sort of stopping the immune system from attacking and destroying those beta cells. So while there is the potential to do it, we're not really sure yet how best to proceed with it in order to sort of get the good bacteria that we want and sort of minimize any sort of ongoing immune response that's taking place in the pancreas.
Great, thank you, James. Susan, can you tell us a little bit something about the attempts that have been made to maybe extend the lifespan of this molecule insulin, and what the pros and cons of that might be?
So there are a number of different preparations of insulin. In general, when people take insulin to help regulate the blood glucose, there is a longer acting background insulin. And though the current insulins last between 16 and 24 hours, and those help to maintain a steady level that is important, and that in a pump is operated by constant, very small amounts going in. The hope is that there would be really good long-acting insulin to maintain that, and some of our newer insulins are very much better than those that would have been available 20 or 30 years ago. The thing is that you can't use long-acting insulin and only long-acting insulin, because the increased load on the metabolism that eating causes requires something that can work very quickly. And so, generally speaking, for the moment, we would always need both a short-acting and a long-acting insulin. Ultimately, there's some interest in something called a smart insulin that might be able to work only when the blood glucose is high. That's still very much at an experimental level, and I believe that would be a long-acting insulin and a very exciting advance if it were to be available.
Right. Well, thank you both. I'm conscious of time passing, but thank you once again, indeed, for two superb presentations this evening. All the very best with your research, and for World Diabetes Day on Sunday as well. So, thanks for all our attendees this evening. I'd just like to call upon my colleague now, Karen Edwards, who's just going to capture some feedback for us. But I'd also like to briefly mention as well that the next talk in our series will now not be until January, the 13th of January, 2022, and we have Professor Frank Kelly from Imperial College speaking on air pollution and public health. How is the UK doing outside the EU? So that's Thursday, the 13th of January. We hope to see many of you again back on that date. So over to you, Karen.
Thank you. Thanks, James. And thanks to Susan and James for an excellent presentation this evening. And as always, we're very keen to get your feedback. So if you wouldn't mind, if you have a phone or another device, maybe you could just log into menti.com, and it'll ask you for a code, which is here on the screen, 61557563. Okay. So great. Okay, getting some people logged in. So if you can tell us first of all where you're joining from today, it'd be really good to know. Okay. Welcome to our Scottish audience and our English audience, as well as our obviously predominantly Welsh audience. Great to see you. Thank you. Okay, so while you're completing that, I'll just move on to the next slide. Yes, you know the score now. Have you attended before? Yes, quite a few. Good to see so many new members at our lecture too. Thanks very much. Okay, so where did you find out about this evening's lecture? Okay, various ways there. Recommendation from teachers, so I'm guessing we have a lot of school pupils in this evening, which is great. Okay. So why did you attend the lecture? Just select the top 2 reasons. Okay, mostly to be informed, and I'm sure we'll all agree that we have been informed this evening. Thank you. And as a result of viewing the lecture, will you go away and maybe do some more research, or do you feel that your knowledge of the subject matter has improved, which looks like most of you feel that way, which is great. Okay. And then finally, if you could just give us 3 words to describe your thoughts on this evening's lecture. That's great. Fascinating, interesting, informative, lovely. That's great. Thanks for that. Thanks for your feedback. Okay. I'll stop my share, and I'm not sure if James wants to say anything else, but all that's left to say, I believe, is we'll see you in January, hopefully, at our next lecture. Hard to believe I'm going to say this, but it's Happy Christmas and Happy New Year to you all.
Stay safe, everyone. Good night.
Good night. Thank you.